Hello everyone. In our previous lectures, we talked about what is meant by drug discovery, what is medicinal chemistry. We talked about what is meant by the term drugs. We talked about the term targets, and we also talked about druggable targets, right? And with those basics in mind, today we'll talk about drug design or rational drug design or ligand design or NCE design. Okay. Uh, just a moment. I am not able to hide this thing. Anyways, yeah. So, drug design or rational drug design or ligand design or NC design. NC is nothing but novel chemical entity. Many people refer to it as new chemical entity as well. All right. So, uh, yeah. So what is meant by drug design? Drug design, often referred to as rational drug design, or simply rational design, is the inventive process. Okay, it is the inventive process of finding new medications based on the knowledge of the biological target. So here we are talking in terms of reverse pharmacology. Okay, here we are talking in terms of reverse pharmacology. So for what was forward pharmacology? Forward pharmacology, यानी when we know कि a given certain a drug is active and then we try to find out its target. Okay, so that is forward pharmacology. And what is reverse pharmacology? When we know the target and then we try to design or find out or discover a drug against that target. Right. So that is reverse pharmacology. And this definition of drug design is based on reverse pharmacology. So what we are seeing here, it is the inventive process, right, of finding what are we finding? New medications. Based on what? Based on the knowledge of the biological target. Okay. So it is the target knowledge that is already known, and then we are trying to devise or design or discover new medications against that particular target. So drug is most commonly an organic small molecule that we have already seen, right? We had classified the drugs into three different types, and we said that the most widely, type of the most widely seen or observed type of drugs are organic compounds. So more than 95% of the drugs that we use in today's world are organic compounds, right? So the drugs are mostly organic small molecules that activate or inhibit. So they can either enhance or they can either inhibit the function of a biomolecule. So biomolecule is the target of interest, such as protein or enzymes or maybe DNA, RNA, okay, and which in turn results into therapeutic benefit to the patient. So what is the main aim of drug design? The main aim of drug design is therapy. And therapy is what? When the patient is cured, right? So if the given drug is resulting into therapeutic benefit, then nothing better than it, right? And in most basic sense, drug design involves the design of a molecule or design of molecules that are complementary. Okay, now this word we had stressed upon in our previous sessions as well. So we talked about the drugs being complementary. Okay, complementary in terms of shape and complementary in terms of charge to the biomolecular target with which they are going to interact, and therefore the drug would bind to it, right? So for instance, as we had taken this example last time, okay, so if suppose this is a protein and then we want to devise a ligand or a drug against this particular protein, then what we'll do, we'll look into the active site first. Say suppose this is the active site. Okay, this particular site is the active site of the protein. Then we want the drug to bind to this particular active site. So what should we do? We should try and design drugs that are complementary in shape first of all. So if a ligand is shaped this ways, is it going to bind to this target? No. If a ligand is shaped this ways, maybe it is a linear shaped molecule. Is it going to bind to this active site? No. But if we have a ligand of this shape, then the chances of this ligand or this drug binding to the active site would be heightened, right? So these two, as we can see here, this particular part of the receptor and this particular ligand are complementary in shape. Okay, and the other part is, say, suppose there are some positively charged amino acid residues in this part of the active site, and then there are some negatively charged residues in the other part of the active site. So what should we have in the ligand? We should have some negatively charged, functional groups in this part of the ligand, and some positively charged functional groups in the other part of the ligand. So what will happen? There will be ionic interactions, right? And that will lead to binding of the drug to the active site. So this is what is meant by complementarity in shape and complementarity in charge. And only when they are complementary in shape as well as in charge. Now just imagine this site is positively charged, this is negatively charged, right? In the active site. What if if we design a ligand which is complementary in shape but has a positive charge over here and a negatively charged functional group over here? What will happen? These charges will repel, right? So that means complementarity has to be in terms of shape and also in terms of charge, right? So when we design ligands which are complementary in shape and charge, then we say that is drug design. Okay, that is drug design. All right. So let's move ahead. So drug design frequently, but not necessarily, relies on computer modeling techniques. यानी कि using computers, we can try to design newer molecules. We can try to design novel molecules, right? So that is what is mentioned over here. So drug design frequently, but not necessarily relies on computer modeling techniques. So this type of modeling is referred to as computer aided drug design. So when computers are used to assist in drug design, that is called as computer aided drug design. That is the most widely used term. Many times we find the term computer assisted drug design. Okay, so instead of aided, many in some books you will find assisted drug design. So that is also the same. So computer aided, computer assisted drug design. But computer aided drug design is what is the normally used term. So that is what we call as CADD, right? CAD. So computer aided drug design. So computer aided drug design is nothing but application of computers which helps us or assists us in the designing of drugs, newer molecules, NCEs, right? So, drug design that relies on the knowledge of the three-dimensional structure of the biomolecular target is known as structure-based drug design. Okay, so there can be basically two different types of drug design. In one type, we have the structure of the receptor being known to us. Okay, the structure of the target being known to us. So that is called as structure-based drug design. It is also called as receptor structure-based drug design. Okay. So structure-based drug design is nothing but when we know the structure of the receptor. And then the other type can be drug design that relies on the knowledge of the three-dimensional structures of the ligands. Okay. So in the first type, we knew structure of the target. Now we are knowing structure of the ligands, but the structure of the biomolecular target is not known. So ऐसे वक्त पे it is called as ligand-based drug design. Okay, it is called as ligand-based drug design. Okay, once more, I'll just repeat this once more. When we know the structure of the target, but the structures of the are not known, then we call it as structure-based drug design. Okay, but when we know the structure of the ligands, but we don't know the structure of the receptor, then it is called as ligand-based drug design. Okay, it is called as ligand-based drug design. So these are the two fundamental types of drug design. In addition to small molecules, we also have biopharmaceuticals, including peptides and therapeutic antibodies. So nowadays we have so many of those monoclonal antibodies which are being used, right? Recently we had, I think, two monoclonal antibodies that have been approved for the use in Alzheimer's disease. So in Alzheimer's disease, we all know, for ages, almost like 20, 30 years, there have been no new drugs that have been discovered, right? But recently, two different monoclonal antibodies have been approved for use in Alzheimer's patient because they reduce the amyloid beta plaque. burden. Right. So similarly, such therapeutic antibodies are also being discovered. So that means biopharmaceuticals are also an increasingly important way of drug design. Okay. So we are talking about small molecules over here, right? When we talk of drug design, computer-aided drug design, initially, older days, may they used to talk about they used to talk about you know only small organic molecules का drug design. Okay, but nowadays we see that in addition to small organic molecules, we also have biopharmaceuticals which are increasingly being discovered. Okay, and this class is now an important class for of drugs, and computational methods for improving the affinity, selectivity, and stability of these protein-based therapeutics have also been developed. So initially computer aided drug design यानी इसका मतलब होता था only design of small organic molecules. But now in today's world we have biopharmaceuticals coming up, and biopharmaceuticals, most of them are proteins, right? So look at this, peptides यानी smaller proteins, right? Antibodies are nothing but proteins, right? So इसका मतलब क्या है कि पहले we used to talk only of ligand and receptor. Okay, we used to talk only about ligand and receptor. Nowadays, we talk about receptor, receptor. In other words, right? क्योंकि receptor यानी क्या protein, ligand यानी क्या small organic molecule. But nowadays we talk about receptor यानी कि protein and receptor यानी कि protein. So we can also write this as protein protein interactions. Right. So initially CAD used to mean only ligand receptor interactions. But now CAD also talks about protein-protein interactions. Why? Because biopharmaceuticals are becoming increasingly important. Okay. So upcoming field. This is the upcoming field in computer-aided drug design. This is almost like a set field. Okay. ये तो पुराने टाइम से चलते आ रहा है, ligand and receptor interactions. Now peptide or protein interactions are what are increasingly being modeled. Okay, and how are they modeled? They are modeled to improve the affinity of one protein to the other protein, or selectivity of one protein towards the other protein, or the stability of these proteins and the stability of these interactions. Okay, so that is what is protein based therapeutics. Okay. Yeah, so let's move ahead. Now, we often use the word, we often use the term uh, drug design. Okay, drug design. And this is actually a misnomer. Misnomer यानी गलती यानी गलत word हम लोग use करते हैं. That is what is meant by misnomer. So the actual term that we should use is not drug design but ligand design. Because what are we trying to do? As we mentioned in the previous slide, see, we are trying to design ligands, right? We are trying to design ligands which can bind very nicely to the receptor. तो हम लोग ने इसको ड्रग डिजाइन कहना चाहिए या ligand based drug design कहना चाहिए या ligand design कहना चाहिए, right? So that's the reason why, you know, the drug design term is often a misnomer. It is actually ligand design. So instead of calling it as computer aided drug design, we should be calling it as computer aided ligand design. Because जब तक ligand does not get approved by the FDA as a drug, we cannot call it as a drug. Okay, so computer aided drug design ये जो term है, ये हम लोग actually output oriented term use करते हैं. Okay, so that means essentially कि if we are trying to design drugs, okay, potential drugs, we're trying to we actually design ligands, but those are potential drugs. So इसीलिए with the expectation that those ligands will go on to become drugs, we call it as computer-aided drug design, right? So what is ligand? Ligand is nothing but a molecule that will bind tightly to its target. Okay, simplest definition of a ligand is a molecule that will bind tightly to its target. And tightly bind कब करेगा? When it is complementary in shape and when it is complementary in charge. Okay, so what are we trying to do when we are using computers For drug design, we are just trying to design ligands which will bind tightly to the receptors via complementarity in shape and complementarity in charge. Okay, so this is the simplest possible explanation of computer aided drug design. All right, so although drug design techniques are used for prediction of binding affinity, and these are reasonably successful. Okay, ये जो मैं कह रहा था कि what is computer aided drug design, it is design of ligands that will bind tightly to the target on account of its shape and charge complementarity, right? And then what do? We try to predict the binding affinity using computer softwares. Okay, using computational softwares. And this is what is done, this is what is the basics of computer aided drug design. Okay, we try to design a ligand, and then we try to see how that ligand would be tightly fitting into the receptor, and then we try to find out how tightly it is binding to the receptor by prediction of the binding affinity. ठीक है, by prediction of the binding affinity. ठीक है, and there are many other properties such as bioavailability that can be calculated, metabolic half life that can be calculated, side effects that can be predicted. ओके, सो ये जो कैलकुलेशन है ना बाइंडिंग एफिनिटी का, बायोअवेलेबिलिटी का, मेटाबॉलिक हाफ लाइफ, साइड इफेक्ट्स एक्सेट्रा एक्सेट्रा, those are all predictions. Okay, and these properties which are predicted must be first optimized before a ligand can become a safe and efficacious drug. So these are the other characteristics which are often difficult to predict with rational drug design techniques. Okay, so usually what we do in drug design, we try to predict the binding affinity, but predicting bioavailability is slightly difficult. Okay, because there can be many factors. As we had talked about yesterday, Lipinski's rule of five ka explanation, the five ways via which the drug can, enter into the cell or cross the barriers that are present in our bodies, right? The five different ways. So all of those ways can come into picture. For example, a drug might passively diffuse into a cell, but it might be effluxed by the efflux proteins, right? So what will happen? That will hamper bioavailability. Okay, so that's the reason why prediction of bioavailability, metabolic half-life, side effects using computer-aided drug design is slightly difficult. Okay, but however, prediction of binding affinity is better. Okay, and that is what we do. So we try to design a molecule that will bind tightly to its target via its complementarity in shape and charge, and then we'll predict its binding affinity. इतना करके हो गया, वो मॉलिक्यूल को सिंथेसाइज कर लो, थोड़ा सा इवेलुएट कर लो, and then give it to the other set of experts, the pharmacologists, the pharmaceutics experts, the analytical experts. Okay, and then what they will do, they will try and find out whether the drug is safe in animals, in humans, whether the drug is amenable to quality control, quality analysis, quality assurance. Okay, and then they will try to find out whether the drug can be formulated into a readily bioavailable form which the patients will find easy to take in, right? So all of that will happen, and only then the drug is going to be marketed as actually a drug, right? So that's the reason why that is what is the basic fund arena wherein within which a computer aided drug design specialist or a medicinal chemist is going to work upon. Okay, the prediction of binding affinity is the most important. बाकी ये सब जो है ना, ये सब फिर फार्माकोलॉजी में वगैरह स्टडी करते हैं, बायोअवेलेबिलिटी, मेटाबॉलिक हाफ लाइफ, साइड इफेक्ट्स एक्सेट्रा. ठीक है. Then, nevertheless, due to high attrition rates, especially during the clinical phases of drug development, more attention is being focused early on in the drug design process on selecting candidate drugs whose physicochemical properties are predicted to result in fewer complications during the development and hence more likely to lead to an approved marketed drug. Okay, so this is what is the difference between medicinal chemistry and pharmaceutical chemistry, as I had explained to you in the very first lecture, right? So this is what is medicinal chemistry. Okay, this is what is medicinal chemistry. Prediction of binding affinity, okay, of a ligand to its receptor. But when we think of the physicochemical properties as well, when we think of the bioavailability, when we think of metabolic half-life, side effects, etc., when we are trying to design a drug, then that entire ambit, that entire arena would be called as pharmaceutical chemistry. Okay? So pharmaceutical chemistry is not only trying to design molecules, but also going ahead and thinking of designing molecules that will actually go on to become drugs by taking into consideration the physicochemical properties, their metabolic half-life, bioavailability, side effects, etc., etc. Okay? And then furthermore, in vitro experiments complemented with computational methods are increasingly used in early drug discovery. So what does that mean? कि एक बार हम लोगों ने ड्रग डिजाइन कर लिया, उसके बाद में वो मॉलिक्यूल्स को सिंथेसाइज कर लो एंड वो मॉलिक्यूल्स को evaluate करो. Okay, so that is what is in vitro experimentation. Okay, so that is what is used in early drug discovery to select compounds with more favorable ADME and T properties. ANA absorption, distribution, metabolism, excretion, and TNA toxicology. Okay, so early on in our studies, in early drug discovery, we need to do this. This is the work of a medicinal chemist or a pharmaceutical chemist. Okay, so do drug design, predict those molecules which will bind very nicely, have good binding affinities, then go on to synthesize those molecules, and then go on to evaluate those molecules so that the molecules can have a favorable ADMET. Okay, ADME, that is what was previously thought of. Okay, आजकल इसको admit बोलते हैं. So ADMET. So toxicology is also very, very important. You know, someone might study just the ADME, but then toxicology is not studied, and then ultimately, due to toxicity, the drug might get, rejected in the clinical trials. So इसीलिए ADMET is what is very, very important to be studied. Now, computer-aided drug design. Now, as I had explained to you all in the previous slide, क्या करते हैं हम लोग? सबसे पहले we'll try to design a molecule or a ligand that will bind tightly to its target on the basis of its shape complementarity and charge complementarity. And then we'll try to predict the binding affinity, right? So this is what we had mentioned. Okay, now the same thing we are going to talk about in the under the ambit of computer-aided drug design. Okay. All right. So the most fundamental goal in drug design is to predict whether a given molecule will bind to its target. Okay. So what do we need to do? We need to predict whether a given molecule will bind to its target. And if so, how strongly? यानी कि we have to predict the binding affinity. In other words, right? So once again, the fundamental goal in drug design is to predict whether a given molecule is going to bind to its target. And if so, how strongly will it bind to its target? Okay, so molecular mechanics or molecular dynamics would be the are most often used to estimate the strength of the intermolecular interactions between the small molecules and its biological targets. Usually, ये जो prediction है of binding affinity, that is done using molecular mechanics, or it can be done using dynamics molecular dynamics simulations. Okay, usually these two are the methods which are widely used for determining or estimating or prediction of the strength of intermolecular interactions between the small molecule and its biological target, and nothing but binding affinity. Okay, nothing but binding affinity. Okay, so these methods are also used to predict the conformation of the small molecule and to model conformational changes in the target that may occur when the small molecule binds to it. Now, say suppose we have one molecule which is going to bind to a receptor. Okay. Now, how do we predict which conformation of the molecule, which three-dimensional arrangement of the molecule is going to bind to the receptor? Right? So we need to model that, and that is also one of the things that is done using computer-aided drug design, using those computational software. So the lowest energy conformer is supposed to be binding to the receptor. Okay, so if we draw a chart, okay, if we draw a graph of the shape of the molecule versus its energy, or rather energy of the molecule versus its binding affinity. So what we find is the lowest energy conformer will tend to bind to the receptor the best. There can be many low energy trots, okay, potential energy curve जो बोलते हैं ना, ये वाला, ठीक है. So this low energy conformer can also bind. Similarly, other conformers which are there along the axis can also bind, but they will not bind properly, right? So it is supposed, it is hypothesized that it is the lowest energy conformer, जिसको हम लोग बोलते हैं global minimum energy conformer. Okay, global minimum energy conformer is supposed to bind to the receptor. So what should we do when we start with our drug design studies? We should always try and find out the global energy minimum conformer of a given ligand. Once we find that global energy minimum conformer, we can predict that yes, this particular global energy minimum conformer is going to bind to the receptor. Okay, so that is also one of the important aims of conformational and changes and conformational dynamics. Study of the conformers which are going to bind to the receptor is also one important aim of computer aided drug design. Ideally, the computational method will be able to predict the affinity before a compound is synthesized. And hence, in theory, only one compound needs to be synthesized, saving enormous time and cost. Okay? So, how did the conventional drug discovery happen? They used to synthesize huge number of molecules. Okay, they used to synthesize huge number of molecules and then evaluate all those molecules. So we know synthesis can happen in a few minutes, or it can happen in a few hours, or it can happen in a few days. And all of those synthesized compounds they used to store in compound repositories. Okay, and then they used to test them each molecule at a time. Okay, so just imagine if, say, suppose one particular molecule is being synthesized in one day. It takes, suppose, one day to synthesize one molecule. If you want to synthesize 1,000 molecules, it will take 1,000 days. And further evaluation के लिए अलग से time. Okay, so this used to be a very time consuming and money consuming effort. तो इसलिए यहाँ पे लिखा है time and cost. But with the advent of computer aided drug design, now instead of trying to synthesize each and every molecule, what the discovery scientists, the drug design people, the drug design experts, what did they start to do? They started to design molecules in silico, यानी कि computer aided drug design software use करके. So they used to do drug design, come up with a few number of molecules, फिर उतने ही molecules को synthesize करो and उतने ही molecules को evaluate करो. ठीक है? So instead of synthesizing thousands of molecules or lakhs of molecules and then evaluating them one at a time, what we can do is we can feed in the data and use computer aided drug design to reduce the number of iterations that are required to reach up to a potent molecule that can be synthesized and evaluated, right? So this is how computer aided drug design saves on time and cost. Okay, now ideally we should be able to design a drug in such a way कि एक ही molecule का एक ही potent molecule design करो and वही molecule का synthesis करो and evaluation करो, and that should be actually the most potent molecule, right? But that does not actually happen. Okay, in reality, the present computational methods are imperfect and provide at best only qualitatively accurate estimates of affinity. So what does that mean? कि हम लोग को we have to synthesize, we have to design more than one drug, and then we have to synthesize those number of drugs, and then we have to evaluate those many number of drugs. But however, it still takes several iterations of design, synthesis, and testing before an optimal drug is discovered. So ideally, this एक ही बार में drug design करके एक ही synthesis करके evaluate करके बस बात खत्म. That should be what ideally computer aided drug design should do. But ऐसा नहीं होता. Okay, drug design करते हैं, उनको synthesize करते हैं, evaluate करते हैं, and then they find out which molecules are more potent, which are less potent. फिर से उस knowledge के basis पे they go on to back to drug design, and then again they will synthesize a few molecules, evaluate a few molecules. फिर से वो data के basis पे they will again go back to drug design, फिर से synthesis, फिर से evaluation. ठीक है. So इसको कहते हैं iteration. Iteration यानी repetition. Okay, so drug design, synthesis, evaluation, these steps are repeated again and again. However, computer-aided drug design has reduced the number of these iterations. तो पहले synthesis करो, evaluate करो, फिर से synthesis करो, evaluate करो, बहुत बार करना पड़ता था. उससे better is drug design, which has reduced the number of iterations that are required. Okay, so computational methods have accelerated discovery by reducing the number of iterations required and have often provided novel structures. So, number of iterations that are required are reduced using computational softwares, and not only that, many times novel structures have been discovered. Okay, many a times novel structures have been discovered. Okay, so that is how computer-aided drug design has actually helped. Okay. All right. So drug design with the help of computers may be used at any of the following stages of drug discovery. The one of the stages could be hit identification. Okay, so hit identification के लिए we do something called as virtual screening. Virtual screening यानी in silico screening that can be done using structure-based drug design or ligand-based drug design. Then computers can be used in hit to lead optimization. So एक बार hit is identified, उसके बाद में we can use it for hit to lead optimization for affinity and selectivity, and that can be done using structure-based drug design and ligand-based drug design techniques like QSAR. And then एक बार lead discover हो गया, तो then we can use that computers for, I mean computational softwares for lead optimization. ठीक है. So lead optimization में we optimize the other pharmaceutical properties while maintaining the affinity constant, or maybe while increasing the affinity, but the affinity should not get reduced. So that is what is lead optimization. So we have all those three steps: hit identification, hit to lead optimization, and lead optimization. In all of these steps computers can be used. ठीक है. Okay, so I think we'll just halt here for a moment, and in the next lecture we'll start with the quantum mechanics and molecular mechanics. Okay, so we were just talking about finding the binding affinity. Okay, we were just talking about finding the binding affinity and how tightly the ligand is binding to its receptor. Correct, but how do we calculate the binding affinity? Those calculations of binding affinity can be done using quantum mechanics and molecular mechanics. Okay, so in the upcoming lecture, we'll talk about quantum mechanics and molecular mechanics.